Hello, my name is Benjamin Fefferman, and I am a Beckman Scholar at the University of Chicago in the laboratory of Professor Xuan He. My current research focus concerns an epigenetic modification in DNA known as 5-hydroxymethylcytosine. In particular, I'm interested in studying this modification in the context of colorectal cancer, and I'm really excited to share my recent research and the implications of this work with you today. In the context of precision oncology, the assessment of biomarkers that are specific for cancer subtype and tissue of origin is essential for the personalized treatment of cancer. This has been increasingly facilitated by recent emergence of next-generation sequencing methods and methods for epigenetic profiling of tumors, both of which have contributed to a new era of tumor biopsies known as so-called liquid biopsies, that, in contrast to traditional histopathological analysis and genomic DNA analysis, rely on so-called circulating cell-free DNA, or cfDNA, to analyze the genome and epitranscriptome and epigenome of tumors. This liquid biopsy method is highly sensitive, which accounts for example, intratumoral heterogeneity. It's minimally invasive, including the collection of a blood sample rather than invasive needle biopsies, for example. It is cost-effective, meaning that one can continually monitor a tumor to assess its progress and evolution, and ultimately, in conjunction with epigenetic profiling, for example, renders improved assessment of disease state and tissue of origin. An exciting possible biomarker that can be analyzed using liquid biopsies is 5-hydroxymethylcytosine, whose chemical structure is shown on the bottom right, involving the attachment of a hydroxymethyl functional group to the 5th carbon of the 6-membered nitrogenous base of a cytosine. 5-hydroxymethylcytosine was recognized as a promising cancer biomarker in part due to recent research that indicates that 5-hydroxymethylcytosine profiling among tumors and among adjacent normal tissue were distinct in both circulating cell-free DNA and genomic DNA. This then led to further results showing a strong correlation between gene expression and 5-hydroxymethylcytosine abundance, with 5-hydroxymethylcytosine generally being associated with active gene expression. In general, however, global 5-HMC levels are lower in cancer cells than in control cells. All of these results combined then to form the so-called 5-HMC classifier, which is an inelastic net regularization statistical method for predicting cancer status and prognosis. Overall, this classifier had higher sensitivity than all traditional biomarkers combined, which was especially significant because it highlighted the role of 5-HMC as a promising cancer biomarker. The classifier that I'm referencing here had highest accuracy among colorectal cancer specifically, and so hence the focus of my recent research on colorectal cancer. In particular, I'm interested in analyzing 4 specific tissue types and cancer status samples or tissue types and cancer statuses in my recent analysis. Specifically analyzing tumor colonocytes, tumor stromal cells, normal colonocytes, and normal stromal cells in pairwise comparisons, namely among normal cells and among tumor cells between colonocytes and stromal cells, and among colonocytes and stromal cells comparing normal and tumor tissue. The goals of this analysis are twofold. One, upon performing dimensional reduction and clustering techniques, I intend to investigate whether 5-HMC profiles are distinct for each of these unique combinations. Second, I'm also interested in first identifying so-called DHMGs, or differentially hydroxymethylated genes, and assessing the biological significance of the genes that are implicated in this differential enrichment analysis. In the top right, to give greater biological context to this system, I've included an H&E staining of a cross-section of the colon in which the epithelium is shown with a unique characteristic and unique epithelial structure with invaginations that form so-called colonic crypts, which interestingly form a unique microenvironment for stem cells and dedifferentiated cells, with a gradient of, for example, Wnt signaling. There are several methods used to prepare raw liquid biopsy samples so that digital sequencing data is produced at the very beginning of my own bioinformatic analysis. In particular, the first method that is used in this analysis is known as nano-HMC seal, which is an experimental method that is a modified immunoprecipitation procedure that employs the beta-glucosyl transferase viral enzyme to transfer an azide-tagged glucose molecule to the hydroxymethyl group of 5-hydroxymethylcytosine. In particular, this azide tag is important because it facilitates so-called click or bioorthogonal chemistry, by which a cycloaddition reaction can be performed between DBCO labeled biotin and the azide tag, so that this biotinylated residue can then be enriched using immunoprecipitation with streptavidin beads, for instance. These streptavidin beads can then be sent downstream for library preparation and next-generation sequencing for deep sequencing of 5-HMC residues. Once next-generation sequencing has been accomplished, my own data analysis methods begin. In particular, I have been learning programming in R, Unix, and Python for creating scripts to analyze RNA sequencing data. In particular, I've been learning to use the high-performance computing cluster, or HPC cluster on campus called Midway, to analyze sequencing data. The computational pipeline that I have used follows the canonical RNA sequencing pipeline, namely starting from so-called FASTQ files, which include raw sequence data followed by quality scores for each base call. I then perform a first round of quality control to assess the sequence quality and sequence content, for example, whether GC levels are too high and general duplication levels. I then proceed to trim NGS adapters from these fragments, which are not necessarily biologically relevant, but are used to ligate adapters to a flow cell during sequencing by the synthesis. Next, I proceed to perform a second round of quality control to assess how well trimming has proceeded during this pipeline. Then I align these fragments to a human genome, a human reference genome, to assign genomic coordinates to these fragments. And then I filter them, which is a procedure for removing PCR duplicates, non-unique mapping reads, or other experimental and digital artifacts from the pipeline. Finally, I perform what is known as peak calling, which is a computational method for identifying regions of 5-HMC enrichment. After peak calling is completed and regions of 5-HMC enrichment are identified, I then proceed with data analysis and visualization in RStudio, as well as gene ontology analysis. In order to better contextualize my own results, it is first important to understand previous results of 5-HMC clustering. My own analysis was performed in genomic DNA. In previous analysis, my research group was interested in understanding 5-HMC clustering in circulating cell-free DNA. In circulating cell-free DNA, previous results indicate that 5-HMC profiles cluster based on tumor status, cancer versus non-cancer, over tissue type. And second, when looking at the clustering from heat maps of this data, the top 50 so-called differentially hydroxymethylated genes showed enrichment in notable canonical cancer-related pathways. For example, regulatory pathways of focal adhesion, as well as PI3K, AKT, anti-apoptotic survival pathways. This again indicates the viability of 5-HMC as a biomarker. My recent results, however, in genomic DNA indicate a slightly contrasting result. First, among the production of a heat map, one can notice that hierarchical clustering in the dendrogram at the top of this diagram shows clustering based on tissue type over cancer status, which is the reverse of what was seen in circulating cell-free DNA. you'll notice that the orange and pink samples are colonocytes clustered together, whereas the and also the light blue and blue stromal cells cluster closer together, while tumor and non-tumor cells appear to be separated. Then I proceeded to perform what is known as principal component analysis, which is an unsupervised method for essentially taking multidimensional data and performing a change of basis such that each axis used to represent the data maximizes the variance of the projection of the data onto each axis, with the restriction that each axis is orthonormal to one another, such that the data is spanned within the vector space. One can notice from this result that my analysis in genomic DNA reinforces the idea that 5-HMC indeed classifies each of these 4 unique combinations of tissue type and cancer status, and it reaffirms the role of 5-HMC as a unique classifier for these factors. Next, I performed principal component analysis among the pairwise comparisons. First, among a comparison of between tissue types, colonocytes versus stromal cells, among normal cells and among tumor cells, and then among colonocytes and among stromal cells between non-tumor and tumor cells. The significant result here is to note that the difference between clusters, the distance between these clusters, is significantly greater in the comparison based on tissue type than based on tumor status. Finally, MA plots were produced, noting 3 conclusions and inferences based on the data. One, there appears to be greater differentially hydroxymethylated genes in the top row in the comparison between tumor and non-tumor stromal cells than between tumor and non-tumor colonocytes, which may suggest involvement by the tumor microenvironment or surrounding cells, which influences transcription dynamics and 5-HMC profiling. Second, you'll notice when comparing the top row and bottom row that the bottom row, which is a comparison based on tissue type, stromal cells versus colonocytes, appears to be more enriched in DHMGs than a comparison based on cancer status at the top row between tumor and non-tumor samples, which again appears to reinforce previous results based on clustering that tissue type really is the core of genomic DNA 5-HMC differential profiles. Finally, one can notice that among the bottom row, a comparison based on tissue type, that normal or tumor-adjacent samples appear to have more differentially enriched 5-HMC profiles than tumor cells, which suggests again a possible role for adjacent tissue, possibly the tumor microenvironment. Overall, there are 3 main conclusions from this analysis, namely, 1, from dimensional reduction or PCA methods, there appears to be a distinct separation between each of the 4 tumor type, tumor status, and tissue subtypes, which reaffirms 5-HMC as an adequate biomarker. Second, among hierarchical clustering and heat maps, an interesting contrast was drawn between genomic DNA, in which there is separation based on tissue type, over cancer status, and previous results in circulating cell-free DNA indicating separation based on cancer status over tissue type. Finally, MA plots indicate a significant contribution of possibly the tumor microenvironment and normal tissue towards differential 5-HMC profiling. My next steps in this analysis include gene ontology analysis with a focus on, for example, tumor-associated macrophages, as well as CAFs, or cancer-associated fibroblasts, predictive modeling, as well as performing RNA sequencing data analysis in conjunction with the nano-HMC seal analysis to see if gene expression correlates in clustering and differential expression analysis with the differential enrichment and nano-HMC seal analysis that I present here. Finally, in the long term, this work hopefully can contribute to the development of so-called liquid biopsies for minimally invasive analysis of cancer. Finally, I want to give a huge thank you to all members of the He Laboratory, particularly Diana, my mentor for this research project, as well as my PI, Professor Xuan He, as well as the PI from the Department of Gastroenterology and the Department of Medicine, Dr. Mark Bissonnette. I also want to thank the Beckman Foundation and the University of Chicago for research funding, and thank you to the entire Beckman community for the opportunity to share my recent research today.